dear colleagues, I'm very glad to present for you our today's speaker, Anna Піднебесна from Czech Republic, and she will present to us very interesting topic, explore the human brain. How can we do that? Anna, I give you a speech, so please.
Thank you very much for the invitation and for the introduction. I just wanted to mention that I'm living in Czech Republic, but I'm actually Ukrainian, so that's, yeah. And today I would like to give you just a brief overview of what I'm doing and what we are doing in our group, just with the examples of, just with some overview of the data and of the tasks we are solving, of the questions we are asking, and maybe just a few projects very briefly. So I'm working officially in two institutions. That's the Institute of Computer Science and National Institute of Mental Health in Czech Republic, not in America, obviously. And I'm working under the supervision of Jaroslav Hlinka, who is also, except of being the head of the Department of Complex System and the Institute of Computer Science, he is also leading the COBRA group, which is actually complex networks and brain dynamics group. This young group, it exists less than 10 years, but we are quite a big group already. We have more than 15 people. And here you can see the main topics we are actually working to. So we are working a lot with brain networks, meaning both neuroimaging data analysis and the brain dynamics modeling. Also, we are working, there are several people who are working with causal interaction inference, and we are all working on, in general, complex network analysis, which includes graph theory, some machine learning, and so on. And we sometimes also are doing some applications, not just to neuroscience, but to climate, economics, and so on. But our main interest is actually complex networks in brain. And I will show you mostly my things, and it will be actually neuroimaging data analysis. So we kind of know quite a lot about the brain. We know approximately the brain structure. We know all these things about gray matter, white matter, about the different neuron structure, about the different neuron types, how the signal is spreading, the brain chemistry, and so on. We even kind of know which region in brain is responsible for what activity. That's not completely true, but at least for some easy tasks like some motor tasks or some sensory things, or we of course know occipital lobe, temporal lobe, and so on. But it's really not enough for saying that we really understand how the brain works, and not really enough for sufficient application in life for people with diseases or for some more interesting applications. So what we are doing now, we are doing in our group, and in general now it's like bigger trend that people are doing with not just with the brain structure, but with the brain dynamics. So we are recording the brain somehow and working maybe even on the higher level, not on the level of neurons, but on the higher level with the brain signal. And I will introduce a few methods to record the brain signal and few things, how to kind of, which question we are asking and how we are dealing with the things. So in our group, we are dealing with several neuroimaging techniques, so several ways how to record the brain activity. There are more of them. And of course, every technique has its weak and strong sides. For example, microelectrodes has very good temporal and spatial resolution, but it's super invasive technique, and it's just a very small part of brain can be recorded by this technique. If we go on the other side, for example, for the EEG, which is electroencephalogram, it's kind of scalp thing, so that's just the head, which scientist or the doctor puts on your head. It's absolutely non-invasive, it's easy, it's cheap, and it has quite good temporal resolution, because since it's non-invasive technique, which is like, since it's EEG, it's measuring the mix of the signal which comes from the brain. And in my talk now, I will actually talk just about two techniques. It's functional magnetic resonance imaging and intracranial EEG. But I will start from showing you several pictures from our different projects, just to give a feeling of what kind of questions we are working on. And for example, we are working a lot on the data that are coming from the epileptic patient who has implanted the EEG inside the brain. So we have some intracranial EEG which are recorded in time, and we ask different questions about how the epilepsy, how the brain behaves in epileptic patient, how the seizure evolves, how what the difference in brain dynamics before seizure, during seizure, after seizure, like before seizure 1 minute, before seizure 1 hour, before seizure 1 week, and trying to ask answer all these questions if we can say something relevant scientifically or even clinically, which is typically very hard and doesn't work, but we are trying. On the similar data, where we have implanted electrodes, we can also ask different questions, because these data are so unique that we are trying to use them as much as possible. And sometimes we are asking those patients actually take part into some experiments. And for example, I was working, one of my last papers was about the visual streams. So we have some hypotheses about visual streams in brain, and taking this data, we want to explore that. Also on this data, I was doing some methodological studies, because it's super important to study how the different techniques can be computed, how they behave on the real data, and how they can be or cannot be transferred from one data set to another data set, even when we kind of assume the type of data is the same, but there are really a lot of math. So these things also should be studied, should be checked, and should be analyzed. Also, we are working with fMRI. So that's a scanner which allows doing many different things. And we are also doing a lot of stuff from the basic, very standard analysis, when we are asking, okay, we have our volunteers who are performing some task. And for example, here it was a visual task. So they were looking at pictures, and we are asking which regions are actually more activated when you're, for example, looking at spiders, more than when you're looking at beetle or something like that. That's a pretty standard thing, but that's not the only question you can ask for this type of data. And my PhD thesis was devoted for the estimation of the neuronal activity from the bold signal, which we are actually measuring during the fMRI scanning. Also, we are working with kind of methodological thing and theoretical approaches with their application on the brain. For